And yeah, then you'll be able to go over any parts that you need to review. Okay, so welcome to those of you who are still joining us into this review session. So I'm going to keep posting the link to the kind of like sign-in form. So you only need to fill that out once. I'm going to keep posting it because people kind of trickle in into the session. So if you have already filled it out, then there's no need to fill it out a second time. And then I'm going to share my PDF of the lab exam review packet. So hopefully you have that handy and you've worked on it. Maybe you've attended some of the other review sessions, which is great. It's always good to have, you know, multiple exposures to this material because it's tough. It's not intuitive, a lot of this information we're learning. So the more times you kind of go over it, the more likely you are to remember it. So don't feel like you're the odd person out because you know, can't quite remember the details or you're not quite sure about how to explain things, because yeah, this stuff is difficult. So just give yourself credit for even just trying. Okay. Let's see, how many people do I have here so far? 22. All right. So let's go ahead and start with this first page. So this is all concerning exercise 7, which was about the enzymes and bioinformatics. Okay, so what are enzymes? How do we define them? Very simply and straightforwardly. You can either turn on your microphone to answer, or you can just type it into the chat. Either one works.
They are proteins that catalyze reactions.
Exactly, yeah. So they're proteins that catalyze reactions. You can also say that they are catalytic proteins. That would be another way to phrase that. And if they're proteins, then what are they made of? What is the monomer of a protein called?
Amino acids.
Right, yeah, so they are made of amino acids linked together and then intricately folded into some shape. What is their importance? Why are enzymes such a big deal?
That without them, life wouldn't be able to exist. It wouldn't be able to perform its functions fast enough.
Right, so the chemical reactions required for living things just would happen way too slowly to sustain life. So the fact that we have living organisms at all is because of enzymes, because they're able to kind of speed up these chemical reactions. And otherwise, these reactions, they would eventually happen, but kind of like spontaneously and randomly, but they would just take forever to kind of happen on their own. So enzymes, the fact that they can speed up reactions really helps move things along. It helps accomplish a lot in the biological world. So we can put that too. That enzymes accomplish important tasks for living things. So what would happen if you had a really high fever for a long time? Do you think that would affect the enzymes in your body?
Yes, because the two things that can alter the chemical makeup or shape of a protein is high temperatures and or change in pH.
Very good, thank you. Yes, so having a sustained high body temperature, like a fever for a long time, could definitely impact your cellular functioning, because yeah, change in temperature affects enzyme shape, and anything that affects the shape or structure is going to affect the function. So with a very high temperature, some of the enzymes in your body might just start to denature and they won't be as active as they should be.
The weaker bonds that hold the 3D structure won't stay together.
Yeah, exactly. Yeah. So they would denature because, yeah, the weaker chemical bonds, they would just fall apart. They wouldn't be able to hold the structure together. And so without the structure, you're going to lose the function. Excellent. Okay, this next question also has to do with structure, and it's about the specificity of the enzyme. So what makes an enzyme only react with a particular substrate?
The shape of the
the
active
shape?
site.
Excellent. Yeah, shape of the active site. Yeah, the shape of the active site. It's going to be kind of a custom fit for a particular substrate. So only a specific substrate will fit. And so the enzyme might kind of come in contact with other molecules, but it's not going to do anything to them. Those other molecules will just kind of bounce off and not react or not be involved in a chemical reaction because they're not going to fit into that active site. All right, I'm just going to once again share the link for signing in for this session. Okay, let's move on to this question 4.4. The how do enzymes increase the rate of reaction? Is it by providing activation energy or by decreasing activation energy?
Decreasing it.
Yeah, so the answer is going to be A, decreasing the activation energy needed to start that reaction. So it gets you over that kind of energy threshold in order to make things happen. So if you had maybe some isolated enzyme and you froze it, you kept it at 0 degrees Celsius for a long, long time, and then one day you decided to take it out and bring it back up to its optimum temperature, whether that's room temperature or something else, would that enzyme be active again?
Yes.
Okay, why would it still be active if it was previously frozen?
The temperature is lowered, it doesn't denaturate or change the shape, it just slows down the molecules.
Yeah, exactly. So a lower temperature, it does affect activity of the enzyme, but it's not going to affect its structure. So its structure is still intact, and when you bring it back up to its normal operating temperature, it'll still be able to work and catalyze those reactions. And we already talked about some factors that can denature an enzyme. So one of them, of course, is temperature. And then someone else who I'm not familiar with, they mentioned that pH, changes in pH can also affect protein activity or enzyme activity. And what's another one, the kind of third variable? Yeah, salt concentration. So changes in salinity can also impact enzyme function. If you think about, you know, the difference between like a raw vegetable, like something that's fresh and picked off the vine, and a pickled vegetable, that's something that's usually been subjected to and like some kind of brine, you know, and a brine is just a vat of salty water. There might be some vinegar, which has a low pH. So those two things combined kind of help preserve or pickle foods. and it's by denaturing enzymes and kind of halting it from reacting at all. So salt concentration, temperature, and pH. Any questions about this first page? Otherwise, we're going to move on to page 2.
You have a question in the chat.
Oh, I see. Thank you, Chanel. Yeah, is it okay to just say salt?
Professor.
I would still say salt concentration, or you could say salinity, because salinity implies that, yeah, you're talking about salt concentration.
Professor.
Hi, Radek. Nice to see that you're here.
Can you go to the previous page because I was still.
Oh, okay, sure.
Give me just some seconds.
Yeah, so remember we did spend 2 weeks on this exercise 7, so that's why the first 6 pages of this handout are all about exercise 7. And I'm gonna keep this up for, let's say 15 more seconds, and then I'll scroll down to page 2. I want to take a moment to thank everyone for attending. I know it's a beautiful Sunday afternoon outside. I would certainly like to be outside right now. So I applaud you for being here in this review session with me. Oh, good. Yeah. Get that morning hike in. All right. So I'm going to go scroll down now to just at least the top of page 2. So we're talking about what is the optimum pH of an enzyme? So how do we define what that condition is?
I think it depends where it came from.
Yeah, so the optimum pH will depend on where that organism originated from, what their natural environment is. And you could also say that, or you could also define it as, you know, the pH at which this enzyme has the most activity. So why does the pH matter? Why, how does it, you know, affect protein activity or enzyme activity?
It can affect the structure and its ability to catalyze the reaction properly.
Right, so again, we're going back to that structure function relationship. So the pH of the environment can alter structure and therefore function of the enzyme. So if you are messing with that pH, if you are increasing or decreasing it out of that optimum range, you are going to be invariably changing that enzyme. So it's not going to be able to be as active. You're going to lose some of those bonds that hold that, you know, functional structure together. Do all enzymes have the same optimum pH?
No.
No, they don't. So it's going to, again, it's going to depend on where that organism is naturally found. And even if we just take the example of amylase, the enzyme that we talked about in this lab exercise, the human body makes 2 different types of amylase. One is in the saliva and the other one is found, it's secreted by the pancreas. And so one is going to be more active at like a neutral pH, the one in your mouth. And the one secreted by the pancreas is more active at a very low pH. So even within your own body, you could have like the same enzyme, but if it's in different locations, it's going to have a slightly different structure to enable to function at different pHs. Okay, hopefully you have by now, at this point in the semester, become very familiar with the steps of the scientific method. So let's just list those out all together. What do we always start with in the scientific method? Question, yes. Right, and that question, that's our general, what is going on here? Then we create a hypothesis. We could put observation over here. And then, yeah, the prediction. So based on our hypothesis, we make a prediction on what we think is happening. Then we carry out our experiment. First, we have to design the experiment, then we can carry it out. And then finally, we have to kind of explain what we think happened in the conclusion. Yeah. And we can lump results in there too. We can say results and conclusion. Okay, does anyone need these kind of described any further than I have? If there's any step that you're still not sure of, we can take a moment to reflect on it and talk about it. Otherwise, we're going to move on to the next question. All right, so this question, and this is similar to something you might see on the lab exam, is there are going to be a few questions where you are given a graph, some kind of results from an experiment, similar to the ones that we talked about in exercise 7, and you'll be asked to analyze what this graph is showing, like what is the trend, or from this graph, what is the optimal temperature for this amylase enzyme. So it's just a matter of observing the graph and deciding what it's telling you. So here, what it wants us to do is look at the graph at 4 degrees Celsius and at 85 degrees, so the two extremes of temperature on this graph, and then explain what we think is happening with the structure and with the function. So at 4 degrees, what do we think well, we can see what the function is. We have these arbitrary units, these AU, so it's showing us 0.4 something of amylase activity. So what kind of function, how do we describe that?
It's active but reduced energy.
Yeah, so it's active, but maybe we can say low activity. Oops. So there is some activity, but it's far lower than the peak value that is shown in the graph. So there's low activity. If that's the function, then what do we think the structure looks like? So Claude, I keep seeing your box kind of highlight like you're speaking, but I can't hear you at all. So if you are trying to participate, I'm not getting it on my end. So I don't know if there's something wrong with your mic.
I'm sorry.
Okay.
It has the correct structure.
Okay, yeah, so we have the correct 3D structure. It's not. Okay. And that's also going to tell us that it is properly folded. So we know that it's in the right configuration. It's just too cold for it to be at its peak activity that happens in this portion of the graph. So then let's go to the other extreme at 85. So what's going on with the function here?
Inactive.
Yeah, so at 85 degrees, it looks just about inactive. It's, you know, it looks like it's at 0 or maybe just above 0, but basically we're not getting anything from this enzyme anymore. So what does that tell us about the structure? It is, yeah, exactly. It's going to be denatured and unfolded then. So I think the reason why they included this word bank here is when you're describing a graph, like in these answers on the lab exam, we do want you to use certain terminology, certain phrases. So this is just kind of, these are just some phrases to keep in mind when you're taking the exam that, you know, if an enzyme is not showing a lot of activity and it's at a high temperature, then you want to say it's denatured, that three-dimensional structure has unfolded. It's no longer in its active conformation, you know, like using those kinds of buzzwords, that's what your instructors are going to be looking for, that kind of language. All right, so let's move on to the next page. All right, so here we have a hypothetical situation where if we took 1 milliliter of fungal amylase enzyme and added it to a starch solution, keep it at 45 degrees for 15 minutes. And what do we think would be the results if we gave it a Benedict's assay? If we did that test with it, would it be a positive Benedict's result or a negative?
Positive.
Right, yeah, we would expect to get a positive result for Benedict's. And why is that?
Product is maltose, which is a reducing sugar.
Yeah, excellent. So this is bringing us back to one of the very first lab exercises we did with the identifying of biological macromolecules. But remember that Benedict's assay, that was the very first one in that exercise. And the Benedict's tests for reducing sugars. And amylase, when it breaks down starch, it creates maltose, which is a reducing sugar. So if we are, you know, if we're mixing up this amylase with starch, we know we're going to end up with maltose. And so maltose is going to give us a positive Benedict's result. And what about if we took the same mixture and added some DNS? Would we see a color change?
Yes.
Yeah, so yes, we would get a color change. And why is it changing color? What is causing that?
Because the maltose reduces the DNS, and the reduced DNS changes colors to like a darker orange.
Right, yeah, so we are we're reducing the DNS. Oops, not DNA, DNS. Okay, so remember this is a kind of a redox reaction. So the DNS is being reduced. To why do I keep writing DNA? Oh, I guess it's got kind of DNA on the brain. Okay, so DNS is reduced to I forget what that abbreviation was. It's like a different type of DNS. We can just say, well, we know we start with like a yellow solution and then it becomes orange.
ANSA.
It started at 4:30, but I was like screen recording part of it.
Okay, and then maltose is oxidized to, what does maltose become?
Reduce DNS.
That's it.
Not-
Maltonic acid.
yeah, it's going to be oxidized to maltonic acid.
Yes.
Right, so maltose is changing to maltonic acid. The yellow DNS is reduced to its orange colored form with some long chemical name, but that's like the key part of this assay is that color change. And it's happening because maltose reduces DNS. Oh, I guess I can't.
Yep.
I was trying to highlight that. I guess it won't let me. Okay. When we did this enzyme lab with our amylase enzymes from different organisms, why do you think we used that DNS instead of Benedict's? What was the purpose of using this different assay?
The DNS test is quantitative and the Benedict's is only qualitative, so you wouldn't be able to get an amount.
Right, exactly. It comes down to basically how you are measuring this. So Benedict's assay is great. It's very simple and quick result, but it only tells you that maltose is there. It doesn't tell you how much of it is there. So it's qualitative. It just gives you like a yes or no answer, basically. And the DNS assay is quantitative. And how do we quantify the amount of DNS using, or I'm sorry, how do we quantify the amount of maltose using DNS? What's that tool that we used?
Is it the spectrophotometer?
Yeah, exactly. We measured, we were able to measure the amount of maltose or estimate the amount of maltose based on absorbance using the spectrophotometer. Okay, so if we took this mixture that we talked about at the very top of this question and then perform the Biuret or puree assay, we would get a positive result. Why would it be positive? What is causing that result?
Purette test for protein.
Right, so the Biuret assay tests for protein. And what is the protein in this sample?
Enzymes?
Right, it's going to react with the enzyme because the enzyme is the protein. Thank you, Chanel, for fielding that question in the chat. One more detail about this Biuret assay. What is it actually reacting with in the protein? How does it detect the presence of a protein?
Peptide bonds.
Yeah, peptide bonds. So those covalent bonds linking the amino acids together, that is what is actually causing that color change. All right, of course, we need to be comfortable with dilutions, hopefully, or yeah, calculating dilutions as well as converting units. So how many milliliters are 350 microliters?
Divide by 1,000.
Yeah, so we would divide by 1,000, which would move the decimal place over 3 places. Right, Bezoit, yeah, so it'd be 0.35 milliliters. And here we're basically doing the same conversion. So we have microliters and we want it in milliliters. So we divide by 1,000 and that would give us 0.5. Okay, now this should look familiar to you, this chart from our amylase activity. So here we have known concentrations of maltose and absorbances that were read in the spectrophotometer, and we need to calculate the concentration of maltose in each tube. So we're going to use our C1V1, C2V2 formula. And add, no, don't want to do that. I want to add a text box. Bear with me. From comments. Oh, no, that's not what I want. Okay, well, whatever. I'm just going to write it in here. Okay, so our C1, that's our starting concentration, and it tells us we have a standard of maltose, that's 5 milligrams per milliliter. So that is our C1. Our starting volume is whatever is in this column. C2, that's what we're trying to figure out. That's our unknown. And then V2, that's our final volume, which is 10 milliliters. Whoa, what just happened? I'm just gonna, okay. Okay, so if we put that all together, we have 5 milligrams per milliliter. Well, for this one, it's times 0 milliliters. Equals C2 times 10 mils. So 5 times 0, that would be 0. And yeah, so here we would have 0 milligrams per milliliter of our maltose because we didn't put any of that standard solution in here. So that makes sense, right? So it might be a little bit easier to do the next one that actually will have a value. All right, so this is how you set it up. And so basically it'll end up dividing this number by 10. So we have 1 divided by 10, which is going to be 1/10 milligrams per milliliter. So each time you're just replacing what goes in the V1 position, because this is our V1 and it changes as we go through our table. So what is the concentration of this solution that has 0.4 milliliters of that standard?
0.2.
Yes, ma'am. It's going to be 0.2 milligrams per milliliter. This one's going to be 0.3. And this one will be 0.4.
Wait, can you say how you got the numbers that you just wrote? Can you say how you calculated those again, please?
Yeah, so up here, basically I'm going to copy what I have written here, except this number is going to be different. So instead of 0.2, in the next row I have 0.4. So it's going to be 5, which is my starting concentration, times 0.4 milliliters of that starting solution, equals C2 times final volume is 10 mils. So we end up, we're going to get 5 times 0.4, which is 2 divided by 10. And that gives us 0.2 milligrams per milliliters.
Thanks.
Yeah, I can write these out too. It's always good to, just practice writing it out, even if you can do it in your brain without writing anything down. It's always good to just give yourself some practice. Typically during the exam, like when we create these questions, they use, nice round numbers to make it easier to calculate. So you can see by looking at all of the list of all of these calculations that the only value that's changing is that V1. So our starting volume is going to change as we go down the rows. And okay, so we have our concentration of maltose and the absorbance as read in the spectrophotometer. And now we have this unknown sample here. So the unknown sample, we don't know how much maltose is in it. All we know is the absorbance. And so if we know the absorbance, then we can extrapolate what the concentration of maltose in that sample is. And for that, you are to make a graph. Not quite sure how to do that with this type of PDF though. Oh, because this is a different version of Acrobat Reader than I'm used to using. So I don't know how to draw stuff out, but basically, oh, this is what someone was telling me to do. Comment. Oh, okay. Thank you, Marilena Papa Georgia. Okay. So let's just crudely draw out a graph. So what's going to be on the x-axis? What's going to be on the y-axis? If we want to graph the information to make a maltose standard curve.
The y-axis has the absorbance.
Right, y-axis is going to have absorbance. Ooh, it's very large. Okay. And then our x-axis.
Maltose concentration.
Right, that's going to have our maltose concentration in milligrams per milliliter. Okay. And we need a title for this. So this is going to be our maltose standard curve. Okay, so if we graph this out, we should end up with about a straight line. So let's see, milligrams, we'll have little lines here.
Professor, we're only going to have to interpret graphs on the test, right?
Right, yeah, you don't have to make graphs. I'm only going through the trouble of making this so that we can use it to interpret the results. I wish I could, ooh, no, I don't want that. Yeah, I know this is taking a while. Okay, we'll just.
I have a question. Do we have a BCR on the exam?
BCR?
Yeah.
I don't know what you're referring to.
Yeah, because I don't know, my professor, he was saying that we might have to write paragraphs talking about what we learned? I don't know if we have to do that for the exam. Like, I'm just asking.
I don't think so. That doesn't sound familiar.
Okay.
So it might have been just your instructor or like something that they are adding to the exam, because we are allowed to like edit it a little bit. So maybe it's something that they've added. All right, so if we had the standard curve graphed and we had our line of best fit, plotted over our points, we know that basically we would use the absorbance to find our concentration. So we'd go here and find the absorbance on the y-axis, and then say it's right here, and then we would draw our horizontal line and then a vertical line down to the x-axis. But let's try to figure this out numerically. So if our absorbance is 0.7, and we see that we had absorbance values of 0.6 and 0.8. Okay, so 0.7 falls just in between that. So if 0.7 absorbance is between 0.2 milligrams per milliliter and 0.3 milligrams per milliliter, what do we think this concentration is that has an absorbance of 0.7?
.25.
Right, yeah, 0.25. So that's kind of a very roundabout way to look at it. So if our absorbance is between these two values, that means our concentration has to be between these two values. And halfway between those two values is 0.25, which if you were to graph this out, that's exactly what the Excel spreadsheet would tell you as well. Okay. Let's see, so we're moving on to the next page. So this is about the bioinformatics exercise, and you will need to, even if you are taking an exam with like the lockdown browser activated, we'll make these specific websites available so you can navigate to them. So there'll be a question or two on the exam where you'll be given a session number. So this is like the specific kind of like catalog number for some enzyme or some protein. And so we'll give you the number and we'll give you the link to the NCBI website. And then we'll ask you, some questions about that protein. So it's up to you to be able to go to the NCBI website, know where to put in the number. Okay, it's going to be up in this like little search box at the top. You want to make sure that you have that protein database selected, and then hit search, and then it'll give you information. And so you might be asked, like, what is the name of this enzyme? Or maybe you'll be asked, from what organism does this enzyme come from? You have to say, oh, okay, this is from Aspergillus oryzae, and like it's alpha amylase, stuff like that. So that's the extent of bioinformatics that you'll see on the exam. So when it asks us here, how do you obtain the Fasta sequence? It's basically just asking you to kind of list out these steps. So the first step is go to the NCBI website. Enter the session number. And then don't forget to select the protein database. Then you're going to obviously click the search button. And if we wanted to get the Fasta from this page, then we would also click the button that says Fasta. So those are the general kind of steps of how you would obtain the Fasta sequence. What is the Fasta sequence? Why do we need it? What was the importance of getting that sequence?
It's easier to analyze information about the protein.
Yeah, so the Fasta the Fasta sequence. Ooh, I really misspelled that one. Sequence allows for further analysis of that protein. What form is this Fasta sequence in? So what is the Fasta actually showing us?
The amino acid chain.
Exactly, yeah, it's going to be, that Fasta is actually the sequence of amino acids. And if you want to get really specific, you can say it's the sequence of amino acids using the single letter codes. So there's one shown here, and it just looks like a string of gibberish, but each letter codes for or represents a different amino acid. So it gives us the amino acid sequence in a format that allows us to further analyze it using other online tools. So that's the purpose of Fasta. It allows us to analyze the protein. So at another, for another question, you might be asked to go to the PepStat website. And again, any website we ask you to go to will provide you like the link for it. And you'll be provided with, you know, access to that website if you're using some kind of lockdown browser. So if we ask you to go to the PepStat website and fill out this kind of information, you want to know kind of where to find that information, where it would kind of be contained. So if you're at PepStat and you enter in the name of some protein, this is what it spits out. And so you want to be able to kind of decipher what this information is.
What was residue mean?
Residue just means amino acid. So by number of residues, it means number of amino acids. So the total number in this one is 478. And then it gives us the kind of different properties of the amino acids, how many of them there are, and then the percentage. So someone asked me in a kind of in a direct message, a really good question that I wanted to share with everyone. They asked, what's the importance of analyzing a protein? You know, like if the purpose of Fasta is to be able to analyze a protein, like, well, what good is that? And what's the purpose of being able to analyze a protein? And kind of like what we did in our lab exercise, we were comparing the same enzyme from 3 completely different organisms. So that's one of the things that we can do with these tools is we can compare, you know, proteins or enzymes from the same organism or from different organisms. We can analyze their secondary structures, you know, like how many alpha helices or beta sheets they have. And, you know, it just allows us to get more insight into them than just looking at an amino acid sequence. Okay, so for filling out this chart, we have to use that PepStat's result on page 7. So how many amino acids are there?
$478.
Yeah, exactly. We've got 478. That's our total number of amino acids. How many of them were polar?
$212.
212. Why? Oh, oops. Wrong box. The non-polar, we had 266. And so we had slightly more non-polar and polar, and then it asks for the percentage, which is also given. You can just kind of round it up, 44.4 and 55.6. Okay.
I just came from the bathroom, so I don't understand like where the numbers came from.
Oh, okay, excellent question. So these numbers are from the PepStats readout, which looks like this. So if you went to the PepStats website and you just typed in, you know, the name of a protein or like gave it the Fasta sequence, this is what it would return as the result. So you need to be able to find the information that was asked for in that table. So one of them is the total number of amino acids, which is shown right at the top here, and it's under residues, or where it says residues, that means like the number of amino acids. And then the polar and non-polar here, it shows you the number of amino acids that are polar or non-polar, and then the percentage. And then that's all we did, was we took those numbers and put them in the table.
I have a question.
Yeah.
Would we have to round up the percentage during the exam?
Yeah, I would just you know, just go to like the first like 1/10 of a percent. I mean, at least that's what I would expect from my students. If they didn't round it, like that's fine too. I would still mark it as correct. On the exam, I believe you have to find the PepStats yourself. Like I think you're given, there's one like kind of longer or like open answer question where you're given a session number. You're given like one of these numbers, and first you have to go to the NCBI site to get the Fasta sequence, the you know, the amino acid sequence, and then you use that at the PepStats site to get this information. So you'll have to know how to like copy and paste and kind of where to find these. I don't think you'll need to know what or how to find the substrate for a protein. Like whatever protein we ask you to look up will give you like that catalog number for it.
I think in Dr. Smith's session, there's some students from another class, I guess, were tested on knowing what the substrate is for a specific enzyme, and he didn't know anything about that, or Dr. Castanos actually, because it was brought up twice. But I think the conclusion was that if we were working with amylase, you should know the substrate is starch.
Right.
That was the only thing that we could figure out to answer that question.
Yeah, that makes sense. Yeah, because otherwise I don't know, yeah, how you would figure that out, like for, at least from this information. I guess it's possible that on, you know, if you looked up on the NCBI site, it might tell you like if the protein that comes up is an enzyme, it might say somewhere in this like long list of information what the substrate is. But I wouldn't expect students to look up that information themselves. So thank you, Chanel, for clearing that up for me. Yeah.
Yeah, it was unique to like one specific class was being asked that. Nobody else knew what was going on.
Okay. Okay. Yeah, I guess that is like the one caveat of having multiple instructors for the same course. Sometimes we teach things a little differently. Okay, so we're going to move on to the next page where it gives us some information about optimal temperature and pH, as well as some of the kind of structural details of these 3 amylases that we studied, human, fungal, and bacterial. But the question actually has nothing to do with this. It asks how amino acids outside the active site contribute to the function of an enzyme.
Maybe they won't like react with the enzyme because the enzyme is specific for that particular substrate.
I think you are on the right track, Radek, but can you speak up a little bit? I didn't quite hear that.
The outside the active site, I mean the substrate spawn react with that enzyme because the enzyme is specific for that particular substrate, so they react with the substrate.
Okay, you're right that, yeah, so outside the active site, the active site is kind of like the business part of the molecule. So the substrate isn't going to bind or react with any other part of the molecule. So you have that correct. But how do the amino acids that make up like the rest of the enzyme, like if the active site, the amino acids there are obviously very important, but what's the importance of all the other amino acids that aren't in the active site? How might they help the function of the enzyme, not just not reacting, but yeah, so Marilena has it, she's onto something. They help with the rest of the structure, they're kind of the scaffold that's holding that active site in its correct conformation. And if you think about, these different organisms and their optimal temperature and pH varies. So the fact that we have these different enzymes that can function in different environments goes back to what their structure is like. So certain amino acids or certain secondary structures are going to make the enzyme more rigid or more fluid and more able to withstand, these environmental conditions. We'll just say they keep. I'm going to try to move this text in a moment. I'm not quite sure how to do that. Okay. I'll move it up here maybe. I don't know. I don't know where would be a good place. Okay, so outside of the active site, amino acids help define that structure and can help maintain that structure if that enzyme happens to be at a very high temperature or maybe at some acidic or basic pH.
Yeah, that's right.
All right, so now we're gonna switch gears and go to the next exercise, which was mitosis. And why can't I? Oh, here we go. So exercise 8 was all about identifying those different stages of mitosis and the cell cycle. So what are the 4 stages of the cell cycle and what's like the main activity in each of them?
Prophase is the first cycle.
So you're talking about mitosis. So the cell cycle includes mitosis, but it also has those other phases too.
G1,
Yeah, so we start with the G1.
S,
Yep. And then G2, and then we also have the M phase, which there we go. And the M phase is where we have mitosis or cell division. So that's what includes the prophase, metaphase, anaphase, telophase, which we'll talk about down here in a moment. So hold on to those thoughts on prophase and the other stages. So what happens in G1? What are we doing in the cell?
Growth.
Right, so we are growing and, maybe replicating organelles. We're basically just making more stuff so that we'll have enough material to split it in half. The S phase, what specific thing happens here?
DNA replication.
Right, so we have our DNA replication. G2,
More growth.
yeah, kind of a continuation of G1. So more growth and preparation for our mitosis, which is, or M phase, which is our mitosis and cell division. I think we might have had some more people join us. So I'm going to post again the link to signing in for this session. So if you haven't already filled that out, please do so. Now, when we move on to the next question, what is the difference between mitosis and cytokinesis?
Mitosis is the division of the nucleus, and cytokinesis is the division of the cytoplasm.
Right, thank you, Chanel. So yeah, mitosis is division of the nucleus, or you could also just say like the genetic material. And cytokinesis is kind of splitting up the cell itself. So cytokinesis divides the cellular material. And cytokinesis looks different in plant and animal cells. So what do plant cells make to split up their daughter cells?
A cell plate.
Yeah, exactly. They make the cell plate. And then animal cells, what is their structure called?
Cleavaging furrow.
Right, yeah, the cleavage furrow. Exactly. So that's the main point there. And why don't they do the same thing with cytokinesis? Why do we have that difference in structure?
Is it because the plant cell's more rigid?
Yeah, exactly. It's the it all comes down to that cell wall. So plants have let's see, plant cells have a rigid cell wall that cannot like pinch in the middle like soft cell membrane that animal cells have. Okay, so it wants you to know the different phases of mitosis and describe what happens in each, as well as being able to identify, from pictures, like what phase a cell is in just by looking at it. And then it also has this interesting question about knowing the numbers of chromosomes and the physical state of the chromosomes in each stage. So actually the number thing we'll deal with in part B. Up here, let's just talk about what state the chromosomes are in. Are they condensed? Are they all loosened up? Are they in a specific position in the cell? So let's try to think about what the chromosomes are, kind of how they appear to us in each stage. So in prophase, what are the chromosomes doing in prophase? What do they look like?
They're condensing?
Yeah, the chromosomes are condensing. So they're actually becoming visible in a microscope. Otherwise, it just kind of looks like one big noodly mess. Becoming visible. In metaphase, what are they doing? Where are they, or what are they doing?
Are they aligning along the center of the?
Yeah, in the center of the cell. So metaphase, they're aligned in the center along that metaphase plate. That's kind of like the imaginary equator of the cell. So they've all met up there.
Sorry, my professor told us not to say metaphase plate. I don't really understand why, but he told us not to say that.
Oh, okay. Well, yeah, I guess if you have if your professor made specific requests for like, you know, what kind of words to use or not use, then yeah, definitely go by that. So I'll just put that in parentheses. So did they say why not to use that, or do they have a reason?
He said why, but I'm not really clear. I think it's something to do with like there's an actual metaphase stage, so I don't know.
Okay. Yeah, so I mean, we all have our like preferences of things. I guess it depends on like how I don't know how familiar we are with these different processes. So yeah, some people get very like nitpicky about how language is used for describing different things, depending on their background. So, okay, what about in anaphase? What are we doing with these chromosomes? What's going on with them?
They're starting to separate, they're being pulled apart.
Yeah, so now the sister chromatids, the kind of duplicates of the chromosome are being pulled apart. Towards opposite ends. That's too many P's. Okay. And then what happens to the chromosomes in telophase? What are they looking like then?
Are they forming their own nucleases?
So yeah, so the new, so within the new daughter cells, we have yeah, like the nuclei are reforming. And then what are the chromosomes themselves doing? What kind of conformation are they going to go into after mitosis?
They start to go to separate ends of the cell. I think the spindles are pulling them.
So they've already gotten to the opposite ends of the cell in anaphase, and telophase is you know, we're already forming the nucleus of each new cell, of each like daughter cell. So basically, what are the chromosomes going to do? Are they going to stay condensed and visible, or are they going to kind of like loosen up?
No, they're loosening up, uncondensed.
Right, yeah. So the nuclear envelopes form around the, I guess we'll just call it the two collections of chromosomes. And the chromosomes are going to decondense, or they're just kind of like going to loosen up, and you won't be able to kind of tell them apart anymore. You can't distinguish between the different chromosomes. They're going to be in a more open conformation. So that's really what I was looking for there, is that their physical state is they have decondensed. So they're no longer packed down into that tight conformation. They've arrived at their destination in the new daughter cell, so they can kind of open up a little bit. So this next question is a little bit difficult at first. If we are not sure what we're doing, so let's go through this together. Oh, Bezoit has a question. Can we find microfilaments in a plant cell? I think you would find them. I think they're still part of like the cytoskeleton. They just wouldn't have a role in making like the cleavage furrow, because we don't have a cleavage furrow in plant cells, but I think you do still have microfilaments. They're just kind of cytoskeletal proteins helping keep that shape. All right. Okay, so here's where we're going to do this. I'm going to have G1. So at G1, we have 46 chromosomes. And are they single chromosomes? Are they duplicates? What is their state?
Duplicate.
So remember G1. So in G1, we have growth and replication of organelles, but we haven't replicated the DNA yet. So they're actually not going to be duplicates. They are going to be, we'll call them single chromosomes or single chromatids. Actually, yeah, I'll just write 46 comma single chromatids. Then in S phase, we know S phase is when we are duplicating. So this is when 46 becomes, we go from 46 single to 46 duplicates. Or we could say sister chromatids. Okay, then in G2, how many chromosomes do we have? Well,
23.
we still have the 46 sister chromatids, right? So that's what where we are prior to mitosis. We have duplicated chromosomes and the duplicates are attached by the centromere. So when we go into mitosis, first we have prophase. We still have the 46 sister chromatids. What about at metaphase? Are the sister chromatids still attached here?
Yes.
Yes, they are. Yeah, so we have 46 sisters. And next we have anaphase. What happens to the sister chromatids in anaphase?
They separate.
So they separate. So we no longer have sisters attached. We have a whole bunch of single chromosomes, right? Because the duplicates are being pulled apart. So what is 46 times 2? How many single chromatids do we have now? Exactly, yeah. So now in anaphase, we no longer have the sister chromatids. We have 92 single chromatids, 46 heading to one end of the cell, 46 being pulled to the other end of the cell. So 92 chromatids total, all being pulled apart. And then when we have telophase, now in telophase we have, formation of our two daughter cells. And so that number has now been halved again. So we at telophase, we have 46 single chromatids in each cell, in each daughter cell.
Professor, I get confused when to use the word chromatid and chromosome.
Okay, so I could have said single chromosome here, and then instead of saying sister chromatids, I could have said duplicated chromosome. So I guess some of these words are kind of interchangeable. I mean, I don't want to confuse you too much, but let's write it out here. So you'd say one chromosome is made up of one chromatid, and then you could have a duplicated chromosome, which is made up of two sister chromatids. So whereas, I mean, I know I use the word chromatids here, but I could have written it out as, one chromosome, and then where instead of sister chromatids, I could have said duplicated chromosomes. So these terms on either side of the equal sign are interchangeable.
Okay, thank you.
Professor, I can't see the rest of part B.
Oh yeah, so sorry, it's I will scroll down. There's like limited space in this box, but
Thank you.
Yeah.
Professor,
Yes.
I think in telophase is still the mother cell. There's no two daughter cell yet, right?
You are right. Yeah, it's not until after cytokinesis that we have the two daughter cells actually formed. So I guess we could say, I mean, another way you could express this would be 92 single chromatids, 46 in each end of the cell, if you wanted to. So one, you know, like this first phrase I have here is more like, oh, sorry, post-cytokinesis. And the other one is like pre-cytokinesis. So yeah, you are technically correct. You still have the 92 chromatids. It's like, it's just that they're now fully separated and in the formation of these two new daughter cells. Shoot, sorry, I keep Xing out of it and then it resets. All right, so we're going to move on to page 9 and then the top of page 10, which has some more mitosis questions. And I think after that, it'll be a good time to take a break.
Does it help to have some?
So let's get through the rest of these mitosis questions. And in case you're just joining us, or in case you haven't yet filled out the sign-in form, I'm going to post that again to the chat. So make sure you have filled that out. All right, so for the mitosis lab, where we looked at cells from an onion root tip and a whitefish blastula, why were we using these specific tissues to look at mitosis? Right, Leanne, thank you. So it's because their cells are constantly dividing. So these are tissues that were actively developing. So all of these cells were in different stages of mitosis. And when cells undergo mitosis, what is the kind of genetic makeup of them? Or how do the genetics of the daughter cell compare to, you know, the parent cell?
They're the same?
Yeah, they're the same. So genetic consequences, I feel like that phrase is a little bit misleading. So I would say like the genetic result maybe of mitotic cell division. So the daughter cells are genetically identical to the parent cell and to each other. And now let's list some basic uses for this mitotic cell division. What is mitosis used for, either by single-celled organisms or by multi-celled organisms like us?
Tissue repair.
Production?
Yeah, I think I have asexual reproduction for single-celled organisms. Tissue renewal, yes.
Hello. You
Rose,
going down? Yeah, I'm going down. Hello, hello, hello. Everything okay?
Growth and
Yeah.
development. But yeah, so basically making more of yourself in a lot of different ways. So whether you are repairing tissue or growing, or developing different types of tissues, you're going to need mitosis. And if you are reproducing and you're just a lonely little cell, then again, you're going to need mitosis to make more of yourself. So this is kind of neat because we have like a little drop-down menu for all the different functions. And they're not really labeled with like letters or numbers. So maybe we'll just go from like left to right, starting with the top row, and then we'll go to the next row and the next row and the next row. So this one up here, what phase are we in?
Metaphase?
Yeah, that's going to be a metaphase.
Professor, this is a polar view of the cell, right?
It's an older view. Is that what you said?
No, there's like side view and a polar view.
Oh, right. Yeah, I know. I understand what you're saying. Yeah, so this is going to be like the horizontal or like the side view. The polar view is usually only visible in animal cells. So that would be down here. So typically in plant cells, it's you only ever see the metaphase in one plane. So they're always going to be kind of in the middle, like these little fingers. That's a good question. Okay, so what's happening in this cell here? So we have a couple of different options. Oh, Marilena has told us, yeah, this one's anaphase. So we have the separation of the chromatids and they're at opposite ends. But there's no evidence that we're like forming the two new cells. There's no pinching in. So, this is plant cells. There's no evidence of that cell plate forming. So we can just say that this is anaphase. Okay, what about this next one here?
Prophase?
Or metaphase?
This could be, yeah, so I would say this is more prophase. It's kind of like a subjective call. So whoever said prophase or pro metaphase, what's your reasoning behind that?
I think because the chromatids are more visible and you can see them trying to move about.
Right, okay, yeah, so we can definitely we can distinguish the individual chromosomes. We can see these darkened lines. So, it's probably not interphase, because interphase would be more, like the cell next to it where you cannot tell the different, where, one chromosome ends and the next one begins. Oh, sorry about that. So we definitely know it's prophase, and it's hard to tell if it's moving towards metaphase. It looks to me like there is still some kind of nuclear envelope holding those chromosomes in like that circular space. If this was broken down and if the chromosomes look like they were moving more or like they were repositioning themselves, maybe then I could say pro-metaphase. But I would say—
Okay, thank you.
Professor, on the exam, will there be a picture with multiple ones that we have to identify like this?
Yeah, so on the exam, there'll be a picture and there'll be like 4 or 5 cells that are highlighted and they'll be marked with letters. And the question will say like, what phase is the cell in letter C in? Or, what is occurring to the chromosomes and the cell marked A?
So the ones that they show us shouldn't be ambiguous. It should be easy to distinguish.
Yeah, correct. Yeah, the exam ones are going to be not as hard to label as these ones are.
Professor,
Yes.
how many written responses are there on the exam?
So that's a good question. Actually, I did want to go over like the types of questions to answer. Let's get through these mitosis questions, because then I wanted to take a break, and then before we take a break, I'll go over like the sort of makeup of the exam.
Right.
So can you go back up, please, so I can take a picture?
Yes.
And onto page 8, please.
Oh, page 8.
Yeah, right there. No. I mean page 9.
Okay.
Oh, the top of it.
Okay. This one. Yeah.
Yeah. Thank you.
All right. So let's try to get through the next row. So what phase do we think here?
Pro Metaphase?
Yeah, so I could see Pro Metaphase. This one is another tough one because it looks like we could see individual chromosomes like the previous cell. I think you could argue either prophase or pro metaphase. Again, like whenever I grade exams, you know, I always allow a little bit of leeway when there's that kind of interpretation. But the images that you'll see are not ambiguous. So it's going to be very clear what phase they're in. And I don't think pro metaphase is included on the exam, at least not the one that I've seen. So I guess there is a possibility that your individual instructor might include a question about pro metaphase. I wouldn't, but I can't speak for all of the instructors. Okay, this next one. So here we have, it looks like two little nuclei and a very faint line here.
Telophase.
Yeah.
Telophase and cytokinesis.
Yeah, definitely. So telophase, we got like the creation of our two new nuclei, and looks like there's a cytokinesis that's cell plate forming. So that would be telophase and cytokinesis. This one looks nearly identical. Again, we have like a little faint cell plate forming. Okay. Let's see the next row. This first one here, what do we think?
Interface.
I wouldn't say interphase because you can kind of tell like the individual chromosomes. You know, interphase would be something like this, where you really can't see any like lines, like dark lines of chromosomes.
Anaphase.
This is probably, yeah, like a prophase or a pro metaphase, I would say, because it doesn't look like there's any more like membrane enclosing like that nucleus, and we can tell where the chromosomes are. Let's see, these next two look pretty similar, maybe different stages of the same phase. So what do we have here?
Anaphase.
Yeah, that one's pretty easy. That one's anaphase. So this one, good question, Leanne. What's the difference between prophase and pro metaphase? So pro metaphase is like the in-between step between, you know, the chromosomes just becoming visible and them already being lined up in the center. Like by, you know, whenever we talk about metaphase, they're already there, but they had to have had some kind of travel time, you know, to get from, you know, all bunched up in a nucleus to aligned in the center. So pro metaphase is like a transition phase between becoming visible and being lined up in the center like this. So that's why it's a little bit difficult to identify like what's in pro metaphase. And I mean, metaphase is very obvious. That's an easy one to tell, but the difference between prophase and pro metaphase is kind of blurry. It's not super distinct.
Would you say that pro metaphase has like more darker spots compared to prophase?
Yes, so I would say, yeah, in pro metaphase, the chromosomes are definitely visible, and they're less likely to have like a little like nuclear envelope around them. Like by the time you get to pro metaphase, that nucleus, the nuclear membrane is completely dissolved, and the chromosomes are kind of free in the cytoplasm.
Oh, shit, I need a password.
So let's talk about this next one. So it looks pretty similar to the cell above it, with a slight difference.
600. That ain't that bad, sorry.
What?
So what's the difference between these two cells here?
Cytokinesis hasn't happened yet in the bottom one?
Yeah, exactly. So in this bottom one, I mean, you can tell that there's, you know, like two bunches of chromosomes or genetic material, but there's no line in between them. Like you can tell here, and then also in this image here. So there's no cytokinesis happening yet. And even if you look at like the border of that genetic material, it's kind of rough, you know, it's like we haven't formed an envelope or a membrane around that genetic material yet. So we're definitely in this one still in telophase, no cytokinesis happening yet. We're still, you could even make an argument that this is like very late anaphase, but luckily that's not an option. Okay, so let's see, I wanted to, so a lot of them look very similar. I wanted to talk about this one because we haven't had this phase yet. What phase is this cell in? We cannot distinguish individual chromosomes. It looks like everything is contained in a nucleus still. So what phase would this be? Yeah, this would be interphase. Kind of looks like a bowling ball because it's got like 3 dark spots. But yeah, this would be, you know, we're ready for mitosis. We haven't started it yet. So any of these cells where it just looks like a kind of fuzzy orb in the center of the cell, it's probably interphase. So we haven't started any of these mitotic activities yet. All right, unless anyone has some objection, I'm going to move on to the next page. If there's one of these images that you really want to discuss or ask about, then feel free to bring it up. Otherwise, I'm going to move forward.
Professor, what's that one in the very far bottom right corner? Sorry, left corner. Sorry, left corner.
Left corner.
Is that prophase?
Yeah, I would say this is prophase, maybe pro metaphase.
Okay, thank you.
And we had a question that, so one of these answer choices is anaphase and cytokinesis, that like, what is that? And can we find it anywhere? And I believe that is a like wrong answer that is there on purpose to mess with you. So cytokinesis only happens at the end of telophase. It does not happen at the end of anaphase, or I guess it can potentially in plant cells, but I feel like that is an answer that's meant to just throw you off and make you doubt yourself. Okay, so we have looked at these onion root tip cells. So let's go on to these whitefish blastula cells. I'll try to keep this up here kind of visible in case folks are still looking at it. So these ones are pretty straightforward. So this first cell up here, chromosomes are maybe not as visible as in the onion root tip, but it looks like there's kind of these strands in between 2 clumps of chromosomes. So what stage is this?
Anaphase.
Right, yeah, this is going to be our anaphase, so they're being pulled apart. This one is
Metaphase.
Yes, this is going to be metaphase. What about here? Yeah, that's probably prophase because we can actually see the chromosomes. This cell, it looks like there's absolutely nothing going on in it. So what does that tell us? Yeah, it's going to be our interphase because the chromosomes haven't condensed. It kind of looks like they're not even there. And then this cell, what is going on?
So cytokinesis.
Right, so this is the telophase and cytokinesis. You can see that membrane forming, that little kind of pinching in the center. Exactly. Okay, so as promised, we're going to take a little break. Before we take a break, I'm going to give you some information about kind of like the makeup of this exam. Right. And this is the exam as it was kind of distributed to the instructors, although we, as instructors, we do have some freedom to edit it, or, you know, we can add things or take out things as we wish. So this might not apply to your instructor's exam. Your instructor might choose to change certain things. So don't take this as, you know, the absolute truth. It could be a little bit different, but in general, the lab exam is worth 120 points, and there are 32 questions. You should probably have like at least 2 hours to take it, I think. So there are 16 just general multiple choice questions. Then you have 6 short answers. And, you know, these are the kinds of things where it might ask you about, you know, like the reagent that we used for an experiment, or like one of the questions that we just answered, like, why did we use onion root tip in whitefish blastula for looking at mitosis? Like, that's another possible short answer question. So something that would take, you know, 2 or 3 sentences to answer. You're going to have about 4 multiple choice questions. Oops, sorry, multi-choice questions for identifying mitosis. And then basically you'll have like 5 kind of longer essay questions. 16, 20, right? Oh, right. 6, okay, 6 essays. 6 essay questions where you'll have to maybe do some calculation. So for example, you know, calculating maltose concentration. Interpreting a graph. Interpreting gel electrophoresis results. There's a question about the, you know, P-glow transformation. So let's see, that's 20 plus 12, that's 32. Okay, yeah, so that's like the makeup of the exam. That's how the questions are kind of divided up. Okay, and with that, so it's 5:42 now. Let's take a 15-minute break, stretch out, maybe get something to eat or drink. So we'll be back here at, actually, we'll just make it an even number. We'll be back and starting again at 6 p.m. So my time says 5:42, so that's 18 minutes. We will be back. And I'm gonna leave a little note up here in case someone joins us in the meantime.
Professor?
Mm-hmm.
Could it be possible to go up one page when you're done? I just need to see one last thing.
Sure, yeah.
Professor?
Mm-hmm.
I was wondering, I didn't get time to register.
Oh, sure. Yeah, let me share that link again.
And also your email for to get the recording.
Sure. So I'm actually, once we're done with this session, I'm going to make this into a YouTube video, and I'm going to share that link with all of the professors.
Okay.
So your instructor should get it and post it.
Okay, thank you.
Okay. All right, so next we are going to start looking at exercise 10. This was the last lab exercise I had to do with those molecular biology techniques, so PCR, restriction, enzyme digest, and gel electrophoresis. So these links here go to different videos and interactives that kind of walk you through exactly how these processes work and like what is happening at a molecular level. And they're really, really helpful if you have trouble kind of visualizing like what is going on or what is happening. So if you haven't checked out these links yet, which are also in the lab handouts, then I would recommend doing so because they're really excellently made, and they're made for, you know, with students in mind, that this is something that you can kind of walk through step by step and really see like what is happening at each level. All right, so we're going to start by talking about PCR, or polymerase chain reaction. So we have 3 steps in a cycle. What are those steps and what happens at each one?
Denaturation.
Right, so we start with denaturation. Let's see, I'm going to switch back to black text. Oops, if I can. It'll let me. Oops. So denaturation, at what temperature does that happen?
95?
Yes, so the denaturing, that's the first step, and yeah, so that's gonna be very high temperature. And why do we need such a high temperature? What is actually happening here?
To separate the 2 strands.
Right, so we're basically melting the DNA apart, or separating the 2 strands with very high heat. So we're opening up that helix, and we're going to make way for enzymes to come in and replicate that DNA. So then what is the next step?
Annealing.
Yeah, so the next step is annealing. And what temperature does annealing occur?
45 to 70 degrees?
That's a very big range. So I usually see like 50 to 60. I guess potentially it could be 45 to 70. That's like even bigger window. But usually you see an annealing temperature in like 50s to 60s. I think it could even be like 50 to 65 or something. So that temperature varies on the reaction. And what is occurring at this annealing step?
The primers are binding to the template strands?
Yeah, so with annealing, the primers, the forward and reverse primers, they're finding their specific DNA sequence and binding to it. And we need those primers there in order for DNA polymerase to come and do its thing, because DNA polymerase cannot just put down nucleotides without a primer. That is one of the limitations of that enzyme. So once we have these primers in place, what is the last step?
Elongation.
Right, so elongation, or you might call it extension, either one. So we are kind of making a copy of that segment of interest. And that is going to happen at about 72 degrees, because that is the optimum temperature for that Taq polymerase enzyme. And of course, one of the main things about PCR is that we don't copy the entire genome or the entire chromosome. We're only interested in one short segment of it, whatever that space is between the forward and reverse primers. So it doesn't take very long, a couple seconds really, to extend those segments. So those are our 3 steps: denaturation, annealing, and elongation or extension. So all of these temperatures are all clearly higher than human body temperature, which is about, what, 37 degrees Celsius. So why is it that it's able to perform at such a high temperature? Why don't like the Taq polymerase enzymes denature?
They're thermophilic, and they're from a specific location that's hot in the hot springs.
Right, so the Taq polymerase is isolated from a thermophilic enzyme, or sorry, thermophilic organism found in hot springs. So this organism is used to living at very high temperatures. It's used to operating its enzymes at very high temperatures. So it's not going to denature at 72. It actually has very good activity at that temperature. Okay, I just want to do, sorry. Let's see if I can just change the color here to black. No, I guess not. Okay, whatever. All right, so when we compare regular DNA replication with PCR, what are the differences that we see between, you know, what happens naturally during S phase of the cell cycle and what happens during PCR?
In S phase, you're copying the entire chromosome gene, all of the genes, and then in PCR, you're only copying a specific sequence.
Right, that's one very distinct feature. So in S phase, we're copying the entire genome, every single chromosome. PCR, we're only doing a segment, which by comparison is much, much smaller. What's another difference? How many copies do we make?
In normal DNA replication, you have 2 copies, and in PCR, it's like 1 billion.
Right, so we make 2 copies, or we end up with 2 copies in S phase. In PCR, we make over 1 billion.
Do you have to specify that that's with 30 cycles when you say how many you're making?
No, like when you're just talking about like the differences, when you're doing a compare and contrast of these 2 processes, I would say it's fine to just say you're making lots of copies or making many copies. That is sufficient. Because here it hasn't specified how many cycles there are, so you can't really say for certain how many copies you're making.
Do we need to say that it's 2 to the n power?
I'm sorry, could you repeat that? It's due to
2 to the n power.
Oh, 2 to the n power. Yes. So I think it is, oh, for the next question, yeah, you would want to put 2 to the n power. And then, you know, here it's actually asked you how many copies you would make after 20 cycles. So there you would want to put a specific number. 2 to the n power, where n equals the number of cycles. And I forget what value this comes out to. I think it's over 1 million though. Thereabouts. I don't have the correct type of calculator in front of me. Does anyone have this calculation done?
Yeah, it's 1,048,576. So It's pretty close to a million.
8,000.
576.
576. All right, thank you. Thank you, ladies. Okay, moving on to these questions about the sickle cell trait or the sickle cell allele. So we have these two alleles, the HBA and HBS. How did this HBS allele cause sickle cell disease? So how did the genotype cause the phenotype is really what this question is asking.
A person with a sickle cell disease must have 2 copies of HBS.
Okay, yeah, so we need so 2 copies of HBS required to have sickle cell disease. Yes, so we needed HBS genotype, and then what kind of phenotype does that cause? What happens to the protein that's made?
It's HBS.
So what happens to the hemoglobin made from the HBS allele? How is it different from what's made from the HBA allele?
Because it's mutated?
Yes. Yeah, so we have an amino acid change. Hemoglobin for HBS allele. There's a change in the 7th amino acid of the sequence.
Changes the glutamine to valine?
Right, yeah, so it goes from glu to val, or glutamate to valine. And the significance here is that valine is a hydrophobic amino acid, and so it has this tendency to kind of clump up together, which distorts the shape of red blood cells, causing the symptoms of sickle cell. Causes the hemoglobin to clump, and that distorts the red blood cell shape. So now we have clearly identified how this genotype leads to the phenotype that's associated with sickle cell, which is those sickle-shaped red blood cells. So how are we able to tell the difference between these two alleles?
Because of the restriction site?
Yeah, so which of the alleles had the restriction site?
The normal?
Right, only the HBA, the normal allele, had a restriction site for the enzyme that we used, which resulted in 2 fragments. That could be identified with the gel electrophoresis. Or I guess we could say they were separated. Right, so it was the presence of that restriction site in one allele and the absence of it in the other allele that was allowed us to distinguish between them. So this next question, it shows us 2 primers used in a PCR reaction, and it asks which one is going to have the higher annealing temperature. So how do we figure that out?
With more G's and C's.
Right, so the primer sequence with more G's and C's will have the higher temperature. So if you go through and you count the G and C nucleotides in each sequence, how many are here?
10.
Right, we had 10 G's and C's in this first one. And what about in the second one?
8.
Yeah, we only had 8 in the second sequence, so therefore the first sequence has the higher temperature. All right, so let's talk again about this relationship between genotype and phenotype. So what's the difference between sickle cell disease and sickle cell trait?
Sickle cell disease, the person has 2 copies, and sickle cell trait, he has only 1 copy.
Excellent, yeah, thank you, Rania. So sickle cell disease is 2 copies of that mutated allele. Sickle cell trait is a heterozygote, so they have 1 normal allele and 1 diseased allele. And what kind of inheritance mode is this sickle cell disease?
Autosomal, recessive?
Yeah, it's autosomal because anything that doesn't occur on a sex chromosome is on an autosome. So we know it's autosomal, and it's also recessive because you need 2 copies in order to have the disorder or the disease. So we call that inheritance autosomal recessive. Now we're moving back to PCR and the components that we need. So what are the main ingredients? There's, I guess, 4 main ingredients for a PCR reaction, and what is each one required for?
The template double-stranded DNA.
Right, so we need a template DNA, and the template is what is going to be copied, right? So whatever sample you have, whether it is from a dinosaur bone or a human being or whatever, it could be from a plant. So anything that has DNA could potentially be a template in PCR. So template DNA could be from any source. What else do we need?
Primers.
Yeah, primers. And what's the purpose of the primers?
They extend the strand.
They're not doing the extending. The polymerase is going to do the extending, but the primers do something.
They're the starting points?
Yeah, so they're the starting point for the polymerase. And they also define the boundaries of the segment to be copied. So they tell the polymerase, you know, where to start and where to stop, so it doesn't just go and indiscriminately, you know, copy the whole genome. All right, and then what's another? Yeah. Okay, then we had the Taq polymerase. And this, Chanel, is for the extending of the segments, right? Because once the primers are down, then the polymerase can come in and add in our last ingredient.
The nucleotides?
Right, yeah, the nucleotides. So we need some raw material for, making a new strand of DNA. So we need some extra, A's and G's and T's and C's just kind of floating around in this reaction mixture so that they can be put in the correct order to make into a copy of this DNA segment. Okay, so this had a lot of information on it, this page, and I know this is kind of like the most recent lab exercise that we've done, so I feel like there's more questions about this one than potentially other exercises. So before I move on, is there anything on this page that anyone needs some clarification on or would like to review?
Professor, I can't see the rest in the last question. Sorry.
Yeah, the last question, yeah, because it got kind of cut
Okay.
off. Yeah. Okay. So now we're going to move on, talk about some applications of PCR, and then we're going to get into the restriction endonucleases. I guess I'll kind of leave that up there. All right, so what do we use PCR for? Or we can start with what we used it for in this exercise.
Disease diagnosis.
Yeah, exactly. So disease diagnosis or any kind of like medical diagnosis. So it could be for a genetic disease, could be for COVID. What else can we use it for?
Cloning.
Cloning, sure, yeah. If anyone's into CSI, oh, Bezoit, yeah, you got it. Forensic analysis, exactly. I'm sorry, can you repeat that?
Like testing for paternity?
Yeah, paternity testing, yeah, absolutely. Yeah, all good examples. I know one that was mentioned in the exercise was archaeology. So again, if you had some like dinosaur bones or fossilized eggs and you wanted to kind of analyze that DNA, you could use PCR to help you do that. Okay, so now we're looking at nucleases. So first couple questions are just about what they are. So obviously it's an enzyme because it ends in ase. We have endonucleases and exonucleases. So what's the difference between those two?
Where they cut.
Yeah, exactly. So the difference is where they cut the DNA. So endo cuts within a sequence, and then the exo cuts at the end. So an endonuclease is going to cut within a sequence, but when we add the word restriction in front of it, what does that entail? What's restricted about it?
It decides or it figures out where the specific region is.
Right. So what do we call that specific region? What's the term that we use?
The recognition site?
Yes. Yeah, it only cuts at the recognition site. And it's an endonuclease, so that recognition site has to be somewhere within the sequence. And which one of these restriction endonucleases did we use in our lab? What was the abbreviation for it?
Is it DDE1?
Yeah, I wasn't sure if it was a 1 or a capital I.
I thought it was an I, but Dr. Castano said it was a 1.
Oh, okay. So DDE1. DDE1 it is. And what kind of ends did it produce?
Sticky.
Right, it had the sticky ends or the little overhangs. And typically these recognition sites, they don't always have to be palindromes, but they are often palindromic DNA sequences. What does that mean? How do we define a palindromic DNA sequence?
If you read the top strand from left to right, it matches the bottom strand from right to left.
Yeah, so it reads, I say it reads the same way in the 5 to 3' direction. But yes, you could also say that reading it from left to right on the top is the same as reading it from right to left on the bottom. Top from left to right is the same as. So the complementary sequences, when they are read in the 5' to 3' direction, which is typically, left to right along the top, and then right to left along the bottom, it's going to be identical. So then it asks us to identify the palindromes down here. And the easiest way to do that is to just kind of write in the complementary sequence below and see if it's the same reading from right to left, the complementary sequence. So which one of these are palindromes?
2.
2 is going to be a palindrome, yes.
And 3?
Yeah, 3 is also a palindrome. So we can double check this by, like I said, by just kind of writing it out. Here, I'll use purple. So this sequence would be T, really? TTCCAA. So that's not identical to what we see up here. So this is AAGGTT. The bottom is AACCTT. So that's not the same. This one's going to be CCCGGG. So reading it from left to right on the top or from right to left on the bottom, get the same thing. And this one just says ATATAT, reading it from the other direction, ATATAT. So those two are palindromes. This one is CTTC.
There's a question in the chat. I think
Oh, thank you.
in.
So yes, so when we're talking about a DNA sequence, first of all, DNA is double-stranded. So we have to remember that when it's looking for a recognition site, it's looking for the palindrome to exist with both of those strands, right? It's going to be the same on both strands, reading in the 5 to 3' direction. And it's DNA, so there's no uracil. So when I'm talking about, writing out the complementary strand, it's just, what does the partner strand of this DNA molecule look like? So that's why there's no uracil here. So PCR only has to do with DNA. There's no RNA, so we don't have to worry about uracil. It's just the A's and T's and G's and C's. So this next question asks about how many fragments you can produce, whether you're cutting, linear or circular DNA. So this is something I didn't go over with my students, just out of the interest of time and simplicity, but I think some instructors did emphasize this. So the number of restriction sites means the number of times you're going to cut a specific sequence. So for example, in our lab, this particular enzyme only cut once, but sometimes there might be multiple recognition sites in a given segment of DNA. So you could potentially have, multiple cut sites, and therefore a number of fragments that are produced from that enzyme digest. So if you had a circular piece of DNA and you cut it once, well, all it's going to do is go from a circle to now it's a straight line. So a circular DNA, if you cut once, you end up with one fragment. If you cut twice, you end up with two fragments. So the number of fragments equals the number of recognition or restriction sites. It's a little bit different when you're starting with a linear piece of DNA. If you cut that linear piece of DNA once, you end up with two fragments. If you cut a linear piece of DNA twice, you end up with three fragments. So the number of fragments is actually equal to the number of restriction sites plus 1. Okay. So let's move on to the next page, or I'll leave this last part up so you can still see it. So what is electrophoresis? Something involving electricity, sounds like.
It allows for the DNA to separate through the pores in the gel so they can move based on their size.
Yeah, so yeah, that's kind of the answer to this next one. So what gel electrophoresis allows us to do, that we separate DNA fragments according to size. Oops, I am size.
Does it have to do with the electrodes having negative and positive?
Yeah, so electrophoresis just by itself, it's the technique of using a current, an electrical current, to move molecules. So in our case, we're moving molecules through this mesh of agarose, but you could also create a gel out of different materials. So sometimes it's polyacrylamide, sometimes it's a gel that's like based on, urea molecules. So the gel can really be anything, but the electrophoresis part means you are using electricity to make things move. So that's how I've defined it. And then, yeah, like for this specific electrophoresis that we did in our exercise, we used it to separate DNA fragments according to their size. What is agarose? Chemically, how do we define it or describe it?
Polysaccharide.
Right, it's a polysaccharide, which you can kind of tell because it has that ose ending like most sugars. So it's a polysaccharide, and when you prepare it in a certain way and then it solidifies, it basically creates like a three-dimensional mesh with, lots of holes in it. So yeah, it creates this three-dimensional mesh with tiny pores through which molecules might pass through. So what is going to determine how DNA migrates through this agarose material?
Size.
Right, so the size of the fragment, and that's usually measured in base pairs. And what else?
The charge.
Yeah, charge of DNA, exactly. That's another good one. And again, that's going to depend on the type of buffer you use.
The density of the gel.
Right, yeah, density of the gel, which is usually determined by just how much agarose you have in your mixture. So if it's a really dense gel, then only very, very small molecules are going to be able to be separated. If it's a loose gel, like a Jello jiggler type consistency, then you'll be able to separate larger things. All right. I think that's it for that one. Next question asks about that DNA ladder. It's also sometimes called the marker, but what is the purpose of that DNA ladder?
To use it as a basis for comparison for size.
Yeah, so it's a size comparison reference for our experimental samples. So we can use it to estimate the size of the fragments that we have run alongside it. So it is our standard. So you can call it a reference or a standard. Something against which you could compare your experimental samples. So when we do this restriction analysis, what are some ways that we can use this, as a tool? So what can we accomplish using restriction analysis?
Making insulin.
Yeah, so that was one of the things it talked about in the lab handout.
Identifying mutations.
Yeah, identifying mutations, presence of mutations, or the differences between two individuals. Any other ones?
Drug production.
Yeah, well, that kind of goes with the making insulin.
Oh, genetically modified products.
Yeah. So we can do it. We can use it for cloning or making GMOs. So if you wanted to create an organism or like a bacteria that produces something that normally wouldn't, that you can just kind of splice that gene of interest in. All right, all good examples. Okay, so let's move on to this next one. Here is asking you to identify the enzyme that doesn't make sticky ends. So we're basically looking for which example shows us two arrows that are lining up together at the same spot. And very clearly the answer is SCAI or SCA1. So this enzyme is going to make a blunt end. There's no overhang on the top or bottom. So all the other ones are making sticky ends with just a little piece that's hanging off. Okay, so now it's referring to, at the bottom of page 13, it refers to this example here of the results of a gel electrophoresis. So first it wants to determine the size of the bands in two ways. First, by kind of estimating the size by what it lines up with in the ladder. and then by measuring the distance between the well and the location of the band. And so to do this for A and B, there are these charts all the way down here. So let's see, our DNA ladder, where does it start at 8,000? So here you would, oops, just put the different sizes of the bands from the reference sample, our ladder. And then it wants you to measure the migration distance. So basically, oh, sorry about the whiplash. So basically from this well to this first band, what is the distance in millimeters? Now, I will admit I have not taken a ruler to it myself, so I don't know the exact values.
I have them.
Okay.
Well, we didn't have a ruler, so Dr. Scottus was like, I'm just gonna give you guys the answers.
Oh, okay, that works.
It's 6, and the next one's 8.5. The next one's 12.5, and then 18, 23.
Oh, sorry, hold on. Skip ahead. Okay.
31.5, 38.5, 45.5, and 55.5.
Okay. So now for these unknown bands, I guess it wants you to also measure the distance, and I think it's much more practical to determine the size for the standard curve, to line up kind of where it is and measure it that way. I think that's, I guess, what we're supposed to put in these boxes. So let's start in lane 4, because it's closest to the ladder, so it's easiest to read. So this size is 8,000 base pairs. And it's important to note that this lane contains a sample that is just the DNA by itself. So the DNA, without being cut by any restriction enzyme, is 8,000 base pairs long. Now let's go to lane 1. So A and B, this is where we have the DNA plus echo R1. This is a restriction enzyme. So what are these sizes?
5 and 3.
Yeah, exactly. So we have a 5,000 band and a 3,000 band, and that's good because 5 and plus 3,000 equals 8,000, which is the size of this uncut piece of DNA. So when we add this echo R1, we cut the DNA once into these 2 size fragments. So then in lane 2, we have DNA plus another enzyme called HIND3. So what are the sizes here? So it's not
7,1.
Right, 7,000 and 1,000. Yeah, and again, these 2 values add up to 8,000, which is the size of our uncut fragment. Now, if we have the DNA plus both of those enzymes, we end up with 3 pieces. So what are the sizes of those pieces?
4,000.
Mm-hmm.
2,000.
Yes, ma'am. And then a 1,000. So if we add these numbers up, 4 plus 2 plus 1, wait, why is it? Okay, that equals 7, so that should be 8 though, right? All right, so, oh, but this, yeah, you could have more than one piece at the 1000 mark because this looks a little bit thicker than in D. This band at G is thicker, so that probably means there's more than one fragment that has migrated there.
That's what Dr. Kasana said. She said they were just overlapping each other, so you can't see that there's two.
Okay, yeah. Yeah, so we have cut it, I guess, potentially more than 3 times, but really we just need to look at the results in 1 and 2 to tell us how many times this DNA is being cut. All right, so how many times did we think this was being cut? So when we have the DNA plus the echo R1, we get 2 fragments. And I think it tells us that this is a linear piece of DNA. Right? Yeah, this tells us the linear DNA molecule.
It was cut once.
Yeah, so echo R1 cuts it once. And then what about Hind 3?
It also cuts it once.
Yeah. Once. And so when you add both of these enzymes together with the DNA, you end up with 3 fragments because it's being cut twice. All right. So I'll leave that up for a moment before we move on to the next page. So the next page wanted you to like graph this using semi-log paper, but you're not going to have to graph on the exam. You might have to interpret a graph, but you don't actually have to create one because there's no way we can ask you to do that in the remote exam setting. If we were to graph it, what would be on the x-axis? What would be on the y-axis?
Y is I guess distance traveled.
Right, so y would be distance traveled in millimeters. And then x would be?
Is it the base pair length?
Right, it would be base pair length. So fragment length in base pairs. Yeah, because how far it's going to travel, the piece of DNA travels depends on how long it is, roughly. So it's not going to travel as far. Okay. So it's if it's a very large fragment, it's not going to travel as far. If it's a very short fragment, it will travel far. So basically we're going to end up with like a negative linear relationship for this graph. So it'd be something like that. And then if you knew from measuring up here, if you measured the migration distance for these bands A through H, so let's say band C was, we know it's close to 7,000. So if band C was at like 8.5, then you would say like, okay, distance traveled, and then figure out the fragment length. So kind of how you use the maltose standard curve to figure out maltose concentration from knowing the absorbance of a sample, same principle here. You can figure out the fragment length by how far it traveled through the gel if you have a standard curve. Okay, we are coming back to the sickle cell genotyping. So it's telling us that we have these 2 additional families that were genotyped, so family A and family B, and it asks us to give the genotypes and phenotypes for each parent and child. So for family A, let's see, well, parent 1, I'll just say P1. What is their genotype? So they've got 3 bands here. What does that tell us?
Heterozygous.
Yeah, so they are heterozygote. They have 1 normal allele and 1 mutant allele. And what does that mean for their phenotype?
That they're carriers.
Correct, yeah, carrier. And parent 2, same thing. They have the 3 bands, oops. So they are heterozygous and a carrier of sickle cell. Their children both have the same genotype. They both have the same banding pattern.
They're both normal.
Yeah, they're both normal. Homozygous normal, exactly. Whoop, didn't mean to do that. Okay. All right, so this was family 1. I want to put family 2 over. I wonder if I can do that. Nope. Oh geez, now I don't know what I'm doing. Okay, I just want to
Put a
write down
question in the chat.
How to tell the difference between banding patterns. Sure. Thank you, Chanel, for bringing my attention to that. I keep getting distracted on this screen. So we have the 3 banding patterns and or 3 different bands here, 2 bands here. So remember when we did this exercise that you know, we were comparing the HBA allele to the HBS. So the HBA allele had that restriction site in it. So we ended up with 2 fragments. And the fragments were 233 and 66 base pairs long. So if you see any bands at like a little bit above 200 and then less than 100, you can assume that it is these fragment sizes that you're looking at. So anyone that has these 2 bands, that means that they have that allele, at least 1 copy of that allele. And then the mutant allele had no restriction site. So it did not get cut at all. That segment of DNA that was copied in PCR remained intact. And the size of that segment, which we determined to be just under 300 base pairs. So wherever you see a line at 300, that indicates the presence of a mutant allele. So an individual with all 3 of these fragments, that means they have 1 of each of these alleles. That means they're heterozygous. So these 2 are HBA, HBS. The 2 children, however, they only have 2 fragments. One is at that, you know, a little bit above 200 mark, and one is at the less than 100 mark. So that means the children only have the normal allele. So they are homozygous normal. They don't have the mutant allele at all. So when we look at family B, so parent 1, I'll put this down here. So parent 1 has all 3 bands. So that means they have 1 copy of the normal allele, 1 copy of the mutant allele. So they are heterozygous and a carrier. The parent 2 and then the 2 children, they all have the same genotype because they all show the same banding pattern. They show the 2 fragments, which indicates they only have the normal allele. So the P2, C1, and C2, they are all homozygous normal. So the key here is just keeping in mind that the normal allele.
I gotta do this, so I have to mute them because they're gonna distract me. You know, got to get the people. No.
So the key thing to remember here is that the normal allele was cut into 2 smaller pieces. The mutant allele was not cut, and it's about 300 base pairs long.
But none of them have just that in this. None of them have the disease, I mean. I mean,
Correct.
no.
Yeah, no one is affected here. If they were affected, then they would only have that 300 base pair band. So yeah, the key thing also to keep in mind is that a genotype is made up of 2 alleles. So you can have 2 HBAs, you can have 2 HBSs, or you can have 1 of each. So the heterozygotes have 1 of each. That's why they have a band present at each possible site. So you could always say, okay, it's heterozygous, but then HBA, HBS. Why is it not showing up? That's weird. Okay, there it is. So a heterozygous carrier. Homozygous normal would be just 2 copies of the HBA allele. So this is family A up here, and then I did family B down here. All right, so if we had another couple that was screened for sickle cell trait, it says they had the same banding pattern as the parents in family B. So these 2 individuals, so one is a heterozygote, one is homozygous normal. If these 2 decide to have a child, what is the chance that children will be affected with sickle cell disease?
It's not possible.
Right, so it's a 0% to get sickle cell disease. It's a 50% chance to get sickle cell trait and a 50% chance to be unaffected. And you can figure this out by making a Punnett square. Actually, yeah, I don't know. So if you just have your, so we have one parent who's heterozygous, so they are HBA, HBS. You have another parent that is homozygous normal, so they can only give up the HBA allele, right? And then if you kind of fill in the spaces in between, so we can get an AA child, we can get an AS child, and same thing down here, AA or AS. So having a double mutant, having both of those recessive alleles, that's not possible for this couple. So 0% chance of sickle cell disease. All right, we are coming into the home stretch. So the last exercise we're going to talk about is this bacterial transformation. So this was, here, I'll keep that up for right now. So this was taking that peak glow plasmid and putting it into some E. coli bacteria. So this first question just asks you to match these terms. So we just want to make sure you know what each of these means. Let's see, how is this done? Okay, so LB agar, what number corresponds to the definition of LB agar?
2?
Yes, so LB agar, that's just a general nutrient mixture. It's used to grow any kind of cells like E. coli bacteria, but you can use it for pretty much anything. Okay, what about glycerol? What did we use that for? Yes, so glycerol was actually, yeah, it's used as a cryopreservative because when stuff is frozen in water tends to create these very sharp ice crystals that bust through cell membranes and kind of kill things. But glycerol doesn't do that. Glycerol prevents the formation of these sharp ice crystals. So it's a better way to freeze things without killing them. Ampicillin. What is ampicillin?
Okay.
Right, ampicillin is 6. It's an antibiotic, kills bacteria. Okay, the AMP-R gene.
$12.
Yes, this gene codes for the beta lactamase enzyme. Calcium chloride.
$3.
Yes, calcium chloride, that was what was used to make cells competent or, able to take up foreign DNA. What is beta lactamase? We know it's made from the AMP-R gene, but what does it do? Right, it is number 7. So beta lactamase is an enzyme that breaks down ampicillin, renders it useless. Okay, what is a plasmid?
1.
Yeah, plasmid is that small circular DNA that kind of replicates on its own, does not need the rest of the chromosome in the bacteria to work. How about piglo?
9.
Yes, ma'am. Yeah, piglo is a specific plasmid that has the BLA gene, which confers ampicillic resistance. What is competence? Right, number 4, the ability to take up DNA from the surrounding environment. Okay. It's slightly different from transformation. How do we define transformation?
8.
Exactly, yeah, that would be number 8. So a competent cell is able to transform, or in other words, in order to have transformation, the cell has to be competent. So it cannot uptake DNA without being competent or having that ability. All right, the AMP-R E. coli. So this is referring to specific bacterial cells that have the AMP-R gene.
10.
Right, so the AMP-R means that they are resistant to E. coli. So they're going to create beta lactamase and they're able to grow on ampicillin. AMP-S. By process of elimination, it's going to be number 11. What does that S stand for?
Sensitive.
Yeah, exactly. So AMP-S means it is sensitive to ampicillin, so it will be killed by the antibiotic. Okay, excellent. So I'll leave this up for a moment in case anyone needs to get that down. Okay, so the next page is, I believe, the second to the last page, asking us some questions about this transformation procedure. First one is, what is the link between piglo and ampicillin resistance?
The piglo contains the BLA gene.
Yeah, so piglo contains a gene that confers ampicillin resistance. So it's the BLA gene, or you could also call it the Agar gene. So the pGLO contains that gene, and that can be transcribed and translated into this functional protein by that bacteria, and it gives it the ability to grow in the presence of an antibiotic. So I guess another way to put it would be any bacteria that takes up the pGLO plasmid be resistant to ampicillin. So how did we know that this transformation procedure had worked? What results showed that it worked?
Colonies?
Colonies, and where did these colonies grow?
Do you mean which petri dishes? I'm sorry, what's your question?
Yeah, so how did we know that some of the E. coli had transformed and had taken up the pGLO plasmid?
How do we know they had the pGLO? Because they were glowing green?
Mm-hmm.
From the GFP?
Yeah, so we had bacterial colonies that glowed green under UV light because they had the GFP gene. So that's one feature. And then the other feature they had was what? They grew in the presence of ampicillin because of that AMP-R gene. So the pGLO plasmid did two things for those bacteria, and we used both of those features to identify which bacteria had actually been transformed. So they not only were able to grow and create little colonies on that plate containing ampicillin, but they also glowed green because they produced that fluorescent protein. So that's how we were able to tell that those bacteria were transformed. So how do we know that they didn't come to us transformed? What was the control for this experiment?
We used bacteria that didn't have ampicillin.
Right, so we used, you mean bacteria without the pGLO? Right, or you said bacteria without the ampicillin? So we had samples of just E. coli plus water, right? So it's just plain E. coli, and then we grew them on two different types of plates. So just that regular, not transformed bacteria, the ones that just had water and no pGLO, we put some on just a plain agar plate and some on an agar plate with ampicillin. And those ended up being our two controls. So plate A was our transformed bacteria. We had plate B, which was the E. coli on agar plus ampicillin. Plate C was the E. coli on just plain agar. So what did those controls tell us? What was the purpose of growing just regular E. coli on agar in plate C.
To tell that it was healthy.
Yeah.
Was
Exactly. So that was just to demonstrate that this E. coli, it's functional, it's living, it's does what E. coli is supposed to do in the given environment. So we can also use the word viable. So the E. coli was viable, it was living. And then for plate B, when we had just a regular E. coli on this agar plate with ampicillin added, what did that tell us?
To see if the ampicillin would kill the normal untransformed E. coli.
Yeah, so that was, to demonstrate just the effectiveness of the ampicillin. Because if we had only done plate A with those green glowing bacteria, a skeptic might look at that and say, well, how do we know it didn't glow beforehand? Or how do we know that you actually put ampicillin on that plate and that, how do we know that the ampicillin you used was actually effective? So that's the purpose of those other two controls, to show that just the regular non-transformed E. coli was viable, that it could live and grow and undergo its asexual reproduction. And then we put some on a plate with ampicillin to show that, yes, this ampicillin is effective, it does what it's supposed to do, it can kill regular E. coli. And now we want to detail, again, we want to link the genotype to the phenotype. So what is it about accepting or taking up that P-glow plasmid that allows the bacteria to survive? So if they take up a certain, genetic material, how does that genotype cause the phenotype of survival in the presence of ampicillin? What's the connection there?
The AMPR gene?
Okay, right, so the P-glow plasmid contains the AmpR gene, and then what does that gene create? What's the product
Resistance.
of that? Resistance, but how does it create resistance? What is the protein that results from activation of that gene? Right, Bezoit, yeah, so this creates beta-lactamase. So the gene creates beta-lactamase, an enzyme that destroys ampicillin. So the result of that is bacteria with the plasmid can grow in the presence of ampicillin because they just break it down, they destroy it, so it's not an impediment to their reproduction. All right, the final question, I'll try to keep these few answers up here visible while we talk about this last question. So this is predicting the results from our experiment, or explaining the type of growth we see. So the first plate had our LB agar with ampicillin and arabinose, and our E. coli that was transformed with P-glow.
What's the question?
What kind of growth do we see on each plate?
We should see colonies on there.
Right, so individual colonies on the first plate. What about the second plate where we just have agar?
No growth.
Yeah, we're gonna have no growth at all. And then the third plate with regular agar, regular E. coli.
Bacteria?
Yeah, so I would use the word abundant growth. So here you can't pick out individual colonies. It's literally like covered completely with bacteria growth. So it's not been restricted at all. And likewise, so it says here, you know, you should be able to interpret results of a transformation experiment. That's so if we gave you kind of the reverse of this question, if we showed you a plate and some pattern of growth, you should be able to say, you know, what was probably grown on that plate or what the plate contained. So if it was a circle with a few little dots on it, you could say, oh, those look like colonies. That's probably the bacteria that took up piglo that can now grow on a plate with ampicillin on it or something like that. All right, so this concludes, well, at least the review packet. I'm still obligated to stick around as long as everyone else wants me to. As long, yeah, so you know, if you don't have any further questions or you've gotten what you needed from this review, then of course you're welcome to leave. If you would like to go back to a previous page in case like you joined a little bit later, I can do that as well. Or if you just have other questions about what to expect on the lab exam, I could certainly try to answer those too. So you're very welcome, folks who came. And yeah, thank you for joining me on an otherwise beautiful Sunday afternoon. And good luck on your lab exam.
Professor, someone's asking to see page 3.
Oh, okay. Oh, can I see page 3? Okay, thank you. I was lost in all of the thank yous. Whoops.
Professor?
Yes.
Please, I just wanted to know, are we going to have access to the video, the recording?
Yes, so following this, I'm going to convert the recording into a YouTube video. And then I'm going to send the link out to the other Biology 150 professors. So they will distribute it to you either through an email or on your course page.
So what's going to be the name of the YouTube video, please?
I don't know. It's probably going to be like Biology 150 lab exam review or something like that.
And is it going to be today or some?
I'm going to try to get it today. It takes, because this video is going to be very, very long, it's going to be several hours long. It takes a while for it to upload into YouTube. But like once it's in that format, it's a lot easier to share than like a Zoom recording.
Okay. All right. Thank you, Professor.
Yeah, you're welcome.
Professor.
So do I have any advice for effectively preparing? Well, the questions that you're given here in the review packet they're very similar to what you see on the lab exam. And I also think that the kind of the scope of the questions is also very reflective of what you see on the exam. So, you know, we like to ask questions about like, what did you do in each exercise? Why did you do it? You know, what was the purpose of each reagent or each step? So understanding not just the what, but also the why of each exercise. And then anytime we throw some formulas at you, you can be sure that you're going to have to use that formula in the exam. And typically in exam questions, they don't give you hints. So it'll just be like the text, and it'll be up to you to remember to use C1V1 equals C2V2. So, you know, being comfortable with identifying when to use what formula. I think one of the best ways to prepare is to go back through the lab handouts and read the introductions to them, because there's a lot of good information about, you know, the background of each assay or each activity. And again, like why you do what you do, and the purpose of each part of the exercise. So I think that's a good way to review.
Professor, I was copying the last question, so can you put it?
Yes, yeah, I will go back to the last question. I hope, Ilaris, that you have gotten what you needed from page 3. All right, I'm going to go back to the last page. Was it this one or this one?
No, yeah, the agar, like the circles.
Okay, the agar one. Okay.
And Professor, also, so we have 6 essay questions, right, for the exam?
Yes.
So should we write like a lot for the essay, or should we can write like?
So for by essay question, I just mean it's that it's usually like a multi-part question, and there's just a big text box kind of similar to this one, and it's not necessarily writing like a traditional essay. It's more that, you know, you're asked several questions, some of which include calculation. So it's just kind of like a multi-part question. It's not necessarily like you're writing a formal essay. So don't let that word confuse you. It's going to be something where, you know, there's like part A, part B, part C, and you just have to answer it all in the same text box.
Oh, okay, I get it. Yeah.
Professor,
Mm-hmm.
Professor, can I have page 18? There's something I haven't finished copying.
Yes.
Page 18. Thank you.
Mm-hmm.
That's it. Thank you.
You're welcome.
And Professor, the last part was a little bit more.
Okay.
Professor, are we going to have to calculate genotype percentages and stuff like that?
I think there's one question that's similar to like this question, where it gave you the kind of genotype of the parents, and it asked you to calculate percentage of, you know, children being affected.
Okay, so it's not going to be like a bunch of pluses and multiplications like in a row. It's just like one, like you just like make a Punnett square and figure it out, basically.
Yeah.
Okay.
Professor, sorry for interrupting the last word.
The last
question.
Oh, the last question again. Okay.
I'm almost done.
Okay. Oh, that's a very good question, Leanne. Yeah, it's very, I mean, I think it's good to start with the essay and short answer, because, you know, you definitely want to allow enough time for those, because they tend to be worth the most amount of points. So it's good to, I think, start with those. But yeah, then making sure you have enough time for the multiple choice questions. I mean, I think, doesn't Blackboard show you like how much time you have left, or does it just give you like an indicator at like 10 minutes left or 5 minutes left? I'm pretty sure that it showed you if you were like.
It does it tells you how much time you have. Usually what I'll do is I will like answer like whatsoever off the top of my head, and then I'll go back later and fill in details if I'm worried about time, because then at least I like get what's fresh in my mind on the paper before I forget.
Right, yeah, like going through and just answering like the easiest ones. Like identifying phases of mitosis, that might be easier for some than for others, you know, because it's like a visual thing and if it's straightforward.
I copied everything. Yes, thank you.
Okay.
Page 18, Professor, is it possible to scroll up a little bit?
Yeah, page 18.
Mm-hmm.
Sure.
Thank you.
Thank you, Professor. Have a nice day.
Oh, you're welcome, Radik.
Professor, thank you for tonight. I'll see you on Tuesday. If I have questions, I'll ask you Tuesday. Thank you.
Okay, great. See you, Chanel. Thank you, Professor. Good night. Good night.
Mm-hmm. Professor, I had some little difficulties with mine because my questions end on question 10, and it starts going down, question 9, question 8, and I don't know why, because I saw you had question 14, you had question 2. I don't know. So I just wanted to know, maybe do I have insufficient document?
So I know that yeah, the numbering of some of these questions is kind of off because a lot of these numbers refer to what the exercise numbers for these labs used to be. And so that's why the numbering of these questions is sort of all over the place. But I mean, I'm fairly certain that this is the most recent like review packet. So which we were all the instructors were given like at the same time. So I would assume that if you got it from your instructor this semester, that it's the correct version of, but was it the question, which question number, or like what was the question that you were unsure of?
On enzymes, everything was accurate, but after enzymes, I started having some difficulties figuring out things.
So, okay, enzymes, I think it ended at like page 8.
Yeah, I had that, but here.
Okay, here.
Yeah, here I started having some issues. But okay, I see exercise. So Professor, can you scroll up just a little bit for me, please? Down, sorry, down. Oh, I see. On mine, it says question 7, but yours is question 8. Okay.
Oh, okay. Interesting. That's confusing because I didn't know there's like more than one version of this, unless your professor like, actually, no, I don't know why they would edit it. Okay.
Okay. Then it should be the same thing, but it's just all of it.
So I'm looking at some other folks' comments in the chat. So Leanne, I'm sorry to hear that yeah, you can't go back to questions you previously answered. That makes it really difficult because even in like, you know, a face-to-face exam, you should be able to like go back and like you're able to go back and check your answers. So I think that would be only fair to allow that in the remote exam. So I'm sorry that.
It's an open book, Professor?
I'm sorry, what was that, Corinne?
Is it an open book exam?
No, it's not open book. Yeah.
But you said, were you trying to say that even in class, student can have access to, like, they can go back?
So in the in-class exam, you know, you have like the exam packet in front of you, so you can always like go back and check your answer to like a previous question. But one of the students is saying that their instructor has it set up so like once you answer a question, you can't go back and change your answer. I think it's really challenging. It's a little unfair because like what if you you know, what if you just want to recheck your answer and make sure you put the correct thing like you would in a regular exam? So I think that is maybe a little unfair. So Reem, I see your comment about the bioinformatics page. So the only two websites from the bioinformatics exercise that we really ask you about are the NCBI site. And for this site, like we'll give you one of these numbers, one of these accession numbers, and ask you to, you know, go to the site, and you might be asked to identify, like, you know, what is this protein that goes along with the number we've given you, or what organism, like, does this protein come from, or something like that. So any kind of information that you can get from like this first page, we might ask you about given that accession number. And then for the PepStat sequence, yeah, you might be asked to take a like this type of amino acid sequence, go to the PepStat website, and then if you just hit, you know, search with your amino acid sequence, then it'll spit out this kind of analysis for you. And so the main thing to keep in mind is where it says residues up here, that just means the number of amino acids. So residues equals 478, that means there's 478 amino acids in this protein sequence. You are welcome. Yeah, that's very difficult. So I let my students see the whole exam at once, so they can like scroll up and down. And I guess some instructors don't allow that because they think that allowing the whole exam at once creates more opportunity for academic dishonesty. And I don't necessarily agree with that assessment. So that's why I choose to give my exams differently. But again, you know, each instructor is kind of entitled to teach their class how they want and give their exams how they want. So I'm very sorry to hear that you can't go back and change your answer. So I would just say, yeah, just go through and be confident. Make sure that you've read through like the introduction for all the handouts, the lab handouts, because that will really help you kind of remember what each exercise was for, and like why we did each step, and what each, you know, reagent or chemical was for. Oh, good. I'm glad. You're very welcome, Leanne. You're welcome, Rania. So Nestor, I know you said that you came in late, but is there a specific page you want to look at? Maybe one, or is a specific question that you had questions about?
No, not actually, Professor. I'm just gonna go through the.
Right, just see the answers. Yeah, I'll be posting
I mean,
the
it's
answer.
gonna be the second time of me going through it. I think.
Oh, okay.
Because I attended the other session, so.
Oh, okay.
Yeah,
So
that will kind of help.
Okay. Yeah, like I said, I'll be posting the answer key on the course page shortly, so you can go check that out.
All right, thank you very much, Professor.
Mhm.
Thanks, Professor. I reached out to my professor for the answer key, and he hasn't responded yet. So hopefully he'll get back to me.
Okay. Well, I guess just in case, I'll share my email here, and if you're really, if you don't get it from your instructor, then you can reach out to me and I'll send it to you.
Thank you.
Mhm.